Good morning, everyone. Thank you for having me here as keynote speaker at the International Conference on Environmental Science and Green Energy, or ICESGE 2022, held in Shenyang, China. This conference is sponsored by Shenyang University of Technology and Engineering and Nişantaşı University, Turkey, also by the IEEE Industry Application Society and the ETDIS. My speech today is about characteristics of hydrolytic indigenous bacteria as degradation agent of hospital wastewater, a case study in Central Java. Introduction. Our research was held in Semarang, the capital city of Central Java, Indonesia, targeting two classes of hospital, the B and C, in the Semarang city. Hospital biomedical waste is a source of pollution that has the most impact on human health. It's categorized as hazardous organic waste because it contains pathogenic bacteria, viruses, fungi, toxic chemicals, and radioactive materials. Role of hydrolytic bacteria. Among indigenous bacteria inhabiting biomedical wastewater are hydrolytic bacteria. They have characteristics as follows. First, they can accelerate the degradation of organic matters by producing hydrolytic enzymes. They can suppress the growth of pathogenic microorganisms through food competition. Third, they are significant in reducing pollution parameters, COD, BOD, TSS, and the phosphate in organic waste. Thus, hydrolytic bacteria can naturally reduce the risk of infection and waste contamination. So we need to expand the growth of the non-pathogenic hydrolytic bacteria. Here I'm showing you the SWOT analysis. We have made it. Related with the strength, weakness, opportunities, and threats on our research topic. As seen in this figure, they are factors contributing to the importance of the evaluation of potential of hydrolytic bacteria as bioremediation agent for hospital liquid biomedical waste. Let me start from the general danger of biomedical waste. So this implies that biomedical waste has the risk of infection and their amount of this waste is increased, means the more contamination danger to the society. Problems of biomedical waste treatment in today's world is that first, incinerator is not environmentally friendly. And second, wastewater treatment plant is costly. In Java Island of Indonesia, biomedical treatment issues emerged as issues of coliform, COD, BOD, ammonia, phosphate, and the debit volume case of effluent of hospitals. Bioremediation research on biomedical waste are behind compared to those of other waste, where bioremediation study on biomedical waste in Java has not been found when we started our research. On the other hand, there is high potential of non-pathogenic hydrolytic bacteria to treat biomedical waste because first, they are relatively economical, they are environmentally friendly, and they are able to degrade organic waste. The objective of this study is to understand characteristics of hydrolytic bacteria as degradation agent of hospital wastewater in Central Java, targeting Semarang as the capital city of Central Java province. How to do that? In this slide, I'm showing you the summary of development method of bioremediation agent of hospital liquid biomedical waste using hydrolytic bacteria. The steps were published by our team in Health and Technology Journal. First, samples from primary reservoir of hospital liquid biomedical waste were taken, and then the bacteria were isolated, purified, and selected. After that, the pathogenesis test and strain identification were conducted, followed by phylogenetic analysis to know the kinship of these bacteria. I mean the indigenous bacteria. And then we conducted the bioremediation ability test of the selected non-pathogenic hydrolytic bacteria that meet the criteria of bioremediation agent. And finally, we conducted the microencapsulation of the bacterial consortium in order to preserve the cells of bacteria so they can be stored for a long period of time. Here's more the detailed scheme of steps required to obtain bioremediation agent from groups of hydrolytic bacteria isolated from hospital liquid biomedical waste reservoir and the targeted outcomes of each step. The liquid biomedical wastewater samples came from primary tanks of 2 hospitals, namely Rumani Muhammadiyah General Hospital and KRT Wongsonegoro Regional Health General Hospital in Semarang City. Anti-infection personal protective equipment was used by officers who have been vaccinated in the sampling process and treatment of the bacteria. The selection of hydrolytic bacteria was carried out on skim milk agar, carboxymethyl cellulose agar, and tributylene agar, with positive results marked by the formation of clear zones of hydrolytic enzymes in this figure. The pathogenicity selection was carried out using McConkey and blood agar plates. Next, the antagonistic test was conducted using the overlay techniques here in the corner. And then the 16 rRNA gene amplification was carried out by PCR with bacterial genomic DNA extract as template. Sequence analysis was performed using BLAST-N, and the encapsulation of the selected bacterial consortium was carried out using the freeze-drying method with 4 types of encapsulation materials. First, results of our bacterial isolation is shown here in this table. So this is a table showing us that from the research, 26 isolates of hydrolytic bacteria from liquid biomedical waste were obtained from 2 classes of hospitals. Protease producers here predominate cultivable hydrolytic bacteria isolated from liquid biomedical waste. Okay, and the results were published in Asian Journal of Chemistry in 2018. And then a phylogenetic tree based on the 16 sRNA gene sequences between the selected bacteria resulted 17 isolates belong to the genus Bacillus, Virgibacillus, and related members of this genera or genus. In this phylogenetic tree, Thermococcus gamma tolerans, FG3, was taken as the outgroup or root of the tree. And this result was published in the EET journal, Ecological Engineering and Environmental Technology journal. 6 of 26 selected as bacterial consortium that can be formulated as bioremediation agent. They are mainly from 3 species, Bacillus methylotropicus, strain 2 strains, and then Bacillus amyloliquefasciens and Bacillus licheniformis. And on the right side is the microencapsulation of the 6 strains of the consortium, bacterial consortium. Okay, and then this 6 of 26 bacterial isolates met the selection criteria as described in the research method, so they can be formulated as bioremediation agent. And then the bacterial consortium of bioremediation agents was then encapsulated, and the results was analyzed using scanning electron microscope. And here are the results. We found that the 1-to-1 ratio of encapsulating materials and bacterial biomass can produce quite good results of microencapsulation. We use 4 types of different encapsulating materials. First, Arabic gum, second, alginate, chitosan, and then maltodextrin. We have not published this result, so here I can only leave the question, what are the best bacterial microcapsules? Because the analysis is going on. We arrive at the conclusion. I can conclude that first, the hydrolytic bacteria can be isolated from biomedical wastewater of hospitals. And then Proteus predominates hydrolytic enzyme production among hydrolytic bacteria from biomedical wastewater. 23% of isolated hydrolytic bacterial species are potential to work as consortium in degrading biomedical waste. 1-to-1 ratio of bacterial cells to encapsulant was recommended to create microcapsules of bioremediation agent containing the cells of selected hydrolytic bacteria. And here are the main references of this presentation. And let me end my presentation by stating quotes from Louis Pasteur. Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. Here are the core team of this study. So this is myself from postgraduate program of Clinical Laboratory Science, Universitas Muhammadiyah Semarang. And then Professor Dr. Agus Sabdono, MSc, from Department of Marine Science and Fisheries, Universitas Diponegoro, Indonesia. Finally, I would like to thank you for all of your attention. Stay good and healthy. And thanks to the ICESGE 2022 and all of its sponsors. I hope to see you all again in another occasion.